In order to replicate, the two nucleotide strands of a DNA molecule separate at their base pairs. Each strand then builds its opposite strand by base pairing with free nucleotides. For example, if one nucleotide on a strand has thymine as a base, the free nucleotide bonded to it would be adenine. Each new replicated DNA molecule has one nucleotide strand from the original DNA molecule and one nucleotide strand that has been newly synthesized from free nucleotides in the cell. Voila! 2 DNA molecules from one.
Chapter 12, Section 2, Replication of DNA. DNA replicates by making a strand that is complementary to each original strand. New vocabulary words to be concerned with for this chapter: semi-conservative replication, DNA polymerase, and Okazaki fragments. During semi-conservative replication, parental strands of DNA separate. So we have a parental strand here, and this blue strand separates. One piece goes over here, and a second piece goes over here. These serve as templates and produce DNA molecules that have one strand of parent DNA and one strand of new DNA. In this case, the new is represented by red. This process occurs in 3 main steps: unwinding, pairing, and joining. The unwinding phase, 3 things happen. DNA helicase, an enzyme, unwinds the helix, breaking the hydrogen bonds between the bases as it unwinds. Single-stranded binding proteins keep DNA strands separate during replication. Meanwhile, RNA primase adds a short segment of RNA primer on each DNA strand. The second step, base pairing. The enzyme DNA polymerase, which is right here, adds appropriate nucleotides to the DNA strand from the 3' end. The leading strand is built continuously, while the lagging strand is built discontinuously in small segments called Okazaki fragments. Joining. DNA polymerase removes the RNA primer and fills in the place with DNA nucleotides. DNA ligase links the 2 sections. Know the difference between eukaryote and prokaryote DNA replication?
We talk about it so much. It is the ultimate director for cells, and it codes for your traits. Once you make the new cell, you better have DNA to put in there. I think DNA replication would actually make a great video game. It's actually quite exciting. I'm going to introduce the key players in DNA replication so that you can get some background information. Now, the majority of these key players that I'm going to introduce are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it may be an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act on. So here we go with our key players. First, helicase. This is the unzipping enzyme. If you recall that DNA has 2 strands, you can think of helicase as unzipping the 2 strands of DNA. Helicase doesn't have a hard time doing that. DNA polymerase the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, poor DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer that it makes? It's actually a piece of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that a little later. Now don't feel overwhelmed. We'll go over this sequence in order. Please keep in mind that like all of our videos, we tend to give the big picture, but there is always more detail to every biological process we cover. That's really true for DNA replication. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. There can be multiple origins within the DNA. At the origin, helicase, the unzipping enzyme, comes in and unwinds, unzips, the DNA. SSB proteins, that stands for single-stranded binding proteins, bind to the DNA strands and they keep them separated. Primase comes in and makes RNA primers on both strands. Now remember, that's really important because otherwise when DNA polymerase comes in, it wouldn't know where to start. Now DNA polymerase can get to work. Remember, it's the important enzyme that adds DNA bases. Now you have two strands, right? They're not identical. Remember, they complement each other. They also are anti-parallel. So that means they don't really go in the same direction. With DNA, we don't say a strand goes north or south. The directions for the DNA strands are a little different. We say that DNA either goes 5' to 3' or 3' to 5'. This can seem very confusing. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of the ring structure. Now you do the same thing for the other side, but keep in mind this strand is flipped, just because DNA strands are anti-parallel to each other. So let's count these. Again, clockwise from...
Notice on this left strand for the 5', the 0 is on top, and it's flipped. The 0 is on the bottom.
The oxygen, 1', 2', 3', 4', 5', and the 5' is out of the ring. This strand on the left runs 5' to 3', and the strand on the right here runs 3' to 5'. Well, it turns out that DNA polymerase can only work in the 5' to 3' direction. So the strand that runs 5' to 3' is fine. It's called the leading strand. But the other strand will make it a little tricky. DNA polymerase can only go in the 5' to 3' direction. So RNA primase, it has to set a lot of extra primers down to do that, as shown here. It takes longer too. This strand is called the lagging strand, which is pretty fitting. On the lagging strand, you tend to get these little fragments of synthesized DNA. They're called Okazaki fragments. Amazing name. The primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, remember that's the gluing enzyme as I like to nickname it, has to take care of the gaps in the Okazaki fragments. Now at the end, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call this semi-conservative replication because the two copies each contain one old original strand and one newly made one. Oh, one last thing. Surely you've had to proofread your work before to catch errors. Well, you definitely don't want DNA polymerase to make an error. If it makes the wrong DNA base, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein, or no protein. DNA polymerase is just awesome. It has proofreading ability, which means it rarely makes mistakes. And that's a good thing. Well, that's it for the Amoeba Sisters, and we remind you to stay